disconnected to the audio.
Okay, but you're there now. Yay.
Yes.
All right. So what's been going on with your I guess audio?
No clue. Oh no, my audio was just because I didn't click it yet.
Oh, okay. Well, then what about your Zoom? Like, is it just relegated to Zoom alone, or is it other problems?
In general, everything's slow, but I can see Schoology. Like, I can see all the assignments and stuff, and I can click on the Zoom link, I think. It's like, you know how you click on it to join the Zoom? I can
Yeah.
click on that, and then I get to the post attendance page,
Yep.
like where it says that I've clicked it and that I'm joining, but then the Zoom itself never pops up.
Wow.
It just
Huh.
kind of loads.
Yeah. Well, you've been slow to load the entire semester.
Yeah, I think it's just my Wi-Fi because my dad's also working from home.
Yeah. Yeah. And I'm working from the school because Blake's at home taking final exams this week, and his exams are 3 hours long, and he has to have two programs running. He has to have a program that monitors him as he takes the exam, and he has to have the exam. And then we've got, Patty working from home, me, and I was like, well, I'm not getting kicked off. And the last class I got kicked off, and so I was like, forget it, and I didn't want him to get kicked off either. Okay, let's just begin. So there's a document here. I suppose you probably have seen it. It says Steps to Respiration. You do see that?
Yes.
Okay, and
the folder for today or like yesterday?
Yeah, if a folder for yesterday. Yeah, and so we're going to fill this in. You're not going to fill it in all today. In fact, I'm not sure exactly how much you're going to get filled in today. The goal is by the end of the class on Wednesday, because the next class you guys are presenting. But on Wednesday, we will definitely finish this. So today, all I'm going to deal with is respiration, aerobic respiration, and not lactic acid and alcoholic fermentation. I'm going to save fermentation for Wednesday. We're just going to deal with this first. So you're welcome to, if you've already have this document, you're welcome to fill it in as we go, or if you have it printed. If not, I wouldn't worry about it, and I would just worry about filling it in after class. Nevertheless, first thing I'm going to do is I'm going to play a short video for you. You're going to watch, and then everything that this guy does, I'm going to do it as well, but it's also a good review, just kind of like it being synopsis, and I think it's better that you see it two ways from two different people. But I'm going to break down everything that this guy does in detail. But here we go. We're going to start with this guy first. And I don't think you can. I don't think you can see it, can you?
Is so important.
Oh, I can see the video.
You can see it.
A lot of us have that.
All right. We're not watching the whole thing.
Hi, it's Mr. Anderson. In this podcast, I'm going to talk about cellular respiration. Sometimes students confuse respiration, breathing, just breathing in, and cellular respiration, and they are linked. Cellular respiration, however, is going to take place at the level of the cell, more specifically inside the mitochondria. And so we need oxygen for that. But it's basically taking our food and then breaking it down in the presence of oxygen to make ATP out of it. What if you're a bacteria? Can you do respiration? You sure can. You don't need a mitochondria to do respiration. They can actually use their outer membranes to do aerobic respiration. And so basically, if you are a track athlete, so this is Usain Bolt right here, when you run, you're using respiration to make energy in the form of ATP and allow your muscles to move. And so this is a quick little study I did. I took all of the world records currently right now from the 100 to the 10,000. And so this is, Bekele is the guy who owns the 10,000 meter record. And what I did is I figured out their pace. In other words, how fast they're running. This would be in meters per second for the 100, the 200, all the way down to the 10,000 meter run, which is a little over 6 miles. And what you can see in this graph is that the pace is quickly going to drop off, and then it's pretty much going to stabilize. And if we were to go out to the marathon or continue, basically we're pretty effective at running at a specific pace. But we die really quickly when we're sprinting. And so a way to think about that is the two things we're going to talk about in this podcast is we're going to talk about aerobic respiration. So aerobic respiration is going to be respiration in the presence of oxygen, but we also have almost like a turbo button that is anaerobic respiration. And so if we really need to go fast, we can get that extra speed that we have up here by doing our anaerobic respiration. The problem, if you've ever run, is that when you get out to this 400 meter, you get a buildup of lactic acid, and it's incredibly painful, and so you can't keep that pace going. Example of a lab I did in class was, and it's weird how this exactly mirrors it, what we did was the muscle fatigue lab. So basically you had a tennis ball, and in one hand you had to squeeze it as many times as you could in 10 seconds, and then do another 10 seconds, and do another 10 seconds, and this is the class average. So the class average was, it looks like around 25 times in 10 seconds. But you can see that it quickly drops off, and then it kind of levels off. And so the same thing, this would be that aerobic respiration, and then this is going to be that anaerobic respiration. And it was fun to see the students' faces, because as they start to go anaerobic, your arm just starts to build up of the lactic acid on the inside of it. But before we get there, let's talk about respiration and what it's for. It's for heterotrophs. So we're heterotrophs, and basically what we're doing is we're taking organic compounds in the presence of oxygen, and we're converting that to carbon dioxide and water. What else are we generating? ATP. Now, what kind of things are doing this? Animals, fungi, bacteria are all heterotrophs, and they're using the organic material to actually make energy. Luckily, we have autotrophs like plants and algae, and basically what they're doing is they're converting that carbon dioxide and water back into organic materials. The only thing that's a little deceptive is that plants are also going to break down those organic compounds, and so they do cellular respiration as well. And so everything's doing cellular respiration. It's how we get energy out of our food. Okay, here's our equation. And again, if you know what photosynthesis is, this is simply the opposite. We're going to take glucose in the presence of oxygen. So here's glucose and here's O2. And then we're going to break that into carbon dioxide, water, and then we're going to generate a little bit of ATP. Now, where is the energy sit? The energy sits right here in this hydrogen on the outside of that glucose. And watch what happens to that hydrogen. It's going to fall down and it's going to grab onto the oxygen, because oxygen wants electrons. And so that's where the energy is coming from. And what the energy is used to do is it's used to make ATP. And ATP is that little fuel that we use in all of our cells. This slide is funny, but it's saying this: behold the power of oxygen. So this fire comes from oxygen pulling electrons close to it. And so there's a huge amount of energy found inside that pull of electrons towards oxygen. Now, if we were to do this inside our body, we could get a lot of energy out of our food, but we would also burst into flames. And so we do it in a really controlled process. Just like when we learned photosynthesis and we had to learn the parts of the chloroplast, when you're learning the cellular respiration, you have to learn a few parts of the mitochondria. So first of all, we have these folds on the inside of the mitochondria. Those are called the cristae. And basically what we have is two membranes. We're going to have an inner membrane right here, and then we're going to have an outer membrane right here. And then this space in the middle is called the inner membrane space. And on the inside, mitochondria, we think, used to be bacteria of their own. And so they'll reproduce through binary fission. They have their own DNA. They have their ribosomes. But they are kind of almost living inside us, not as a parasite, but as a symbiont. They're actually helping us as we generate energy. So there are 3 steps in cellular respiration. Let's start with the first one. So the first one is going to be glycolysis. The second one normally we think of as the Krebs cycle. And then the third one is going to be the electron transport chain. And so the first one, I love this diagram here because it's putting glycolysis outside the mitochondria. And so this is going to take place outside the mitochondria. Where would that be? Well, that would be in the cytoplasm of a cell inside your body, or would be right outside of bacteria. But what happens in glycolysis? Basically, we're taking glucose. Glucose is a 6-carbon molecule. And in glycolysis, we're going to break that down into 2 molecules of pyruvate. Each of those have 3 carbons inside it. So the 2 3-carbon molecule, that's what glycolysis does. What do we generate in there? Well, we generate a little bit of ATP. For 1 glucose molecule in glycolysis, we're going to make 2 ATP. The other thing that we make is a chemical called NAD. What we're basically doing is we're transferring high energy electrons to NAD, and we're adding protons to it as well. And we'll get to NAD in just a little bit. Let's follow pyruvate then. Pyruvate is going to diffuse into the mitochondria, and then we're going to have this pyruvate dehydrogenase complex, and basically what it's going to do is it's going to convert that 3-carbon molecule into acetyl-CoA. This is coenzyme A. So basically now we have a 2-carbon molecule. That is going to go into the Krebs cycle. Now, since we're going from a 3-carbon pyruvate to a 2-carbon acetyl-CoA, we're giving off carbon. And that carbon is going to be given off in the form of carbon dioxide. And so when you breathe out a third of that carbon coming out of you is going to come right here from this complex inside the matrix, we call this. I should have said that before. This is the matrix on the inside of the mitochondria. Okay, let's keep watching acetyl-CoA. So it's a 2-carbon molecule. Where does it go next? It goes to the Krebs cycle. And so in the Krebs cycle, we're going to break it down further, and we're going to get rid of these 2 carbons in acetyl-CoA, and we're going to give those off as carbon dioxide. So we're getting rid of carbon dioxide. What else are we producing in the Krebs cycle? You can see here that we're producing a little bit of ATP, 2 ATP, but we're also adding energy, again, to NAD, and we're adding energy to its friend, we'll call this FAD. And so what do NAD and FAD have? They have these high energy electrons, and they're going to carry those electrons to the 3rd step, which is the electron transport chain. Okay, let's get to the electron transport chain then. All of our energy, pretty much, that was in glucose is now in NAD and FAD. So they're going to transfer their electrons, and those electrons are going to go through what's called an electron transport chain. Basically, they're moving through a series of proteins, and the energy of those proteins is used to pump protons. Protons are going to be hydrogen ions to the outside of this inner membrane, into what's called the inner membrane space. So now we've built up all of these protons right here. What happens to the electron? The electron is going to be added to other protons and oxygen that we breathe in, and that's going to make our byproduct, which is going to be water. And so let's slow that for just a second. The oxygen that you breathe is moving in here, and it's going to be that last electron acceptor right in here in the matrix, and we're going to take the protons. What happened to those protons? They'll actually flow through a protein called ATP synthase. Those protons will combine with the electrons and the oxygen, and it's going to make water, which is going to be a byproduct of that. Now, how much ATP do we make down here? Well, we can make around 32 or 34 ATP in this last step. And so in the electron transport chain, we're making a whole heck of a lot of energy. And so it's worth taking a look at how that actually works. So let's go to the electron transport chain. So kind of to situate ourselves, what do we have? Well, we have NAD, our friends NAD and FAD. What are NAD and FAD passing off? They're electrons. Those electrons are going to move through the electron transport chain like this. Every time they go through one of these proteins, it's going to pump another.
And I'm going to pause for a second. So this is going to be the matrix in here. So this is the matrix. And this is the inner membrane space. So there's 2 compartments. Remember, there's an inner membrane, and that separates this space on the outside, which is the, the inner membrane space, and then the matrix. And so you've got 2 compartments, and this is what's so beautiful about this. And this is where I'm literally going to stop here, and then I'm going to go through it myself. As NAD and FAD, as NADH and FADH2 come here, they're going to come along, and they're going to pass off 2 things. One, there's going to be a reaction. There's an enzyme here. You don't need to worry about that enzyme, but that enzyme is going to cleave off this hydrogen, this proton, this hydrogen ion, and it's going to get pumped out here into inner membrane space. But that electron is broken off as well, and it is going to be passing between these proteins. And these proteins are electronegative, and this hydrogen is electropositive. So it keeps dragging tons of this hydrogen over to this side in the inner membrane space. There's only one way for it to get out of there. You get a high concentration of ions here, a low concentration here. And these hydrogen ions will flow through this enzyme called ATP synthase. And it says ATP synthesis, but this enzyme that performs this is ATP synthase. And you just go, the 2 names go hand in hand. But this is just diffusion. Now, why does this ion concentration stay low here? Well, the reason is these electrons, when they come out, they have to get accepted by oxygen. So oxygen is actually broken down by another enzyme. Instead of O2, it's just O. It's just oxygen, free oxygen. It will react with that, that electron and bond to 2 hydrogens that diffuse through here. And then you'll excrete this water. This water will diffuse out through osmosis, and then you'll pee. But you'll keep, this water will keep grabbing, or this oxygen will keep grabbing these electrons and hydrogen to keep this concentration gradient really high here and low here, so that diffusion keeps going. So if you stop breathing, well, then what happens is that this concentration gradient equals out. You stop making ATP, you pass out and die. And so if you ever, get starved of oxygen, well, that's exactly why you fade to black, so to speak, is you stop synthesizing ATP and then your body shuts down. Okay, you may have a question, and I guess I'll go ahead and entertain questions since we're not in any kind of, I guess, fevered pace since it's just you and I. But do you have a question so far?
Yeah. Wait, so this oxygen is coming from like it being broken down, right? This is just from the air?
Yeah, it's just from the air. You breathe it. So this is O2, which is what you breathe. And then, of course, that gets into your blood. It gets into your circulatory system, and then it will diffuse into all of your cells, and then it diffuses into your mitochondria. And then that's where that oxygen is going to come here and pick up these electrons and hydrogen. But yes, this is just from you breathing. That's your only question about this?
I had the most problem with like the electron transport chain just because a lot is going on. But
yeah, there's a lot going on, but I mean, this is what we're looking at, but.
Yeah. Like, so I think it was the Khan Academy, but like as the electrons go through those proteins, do they like get a lower charge or like?
Well, they start to, what happens is that they're going to lose a little energy. This is a very short little chain. And the only way to keep this going is to keep this, whenever this electron here in this fourth complex, complex 4, when it falls out to the oxygen, this becomes a little more electropositive and it keeps dragging these other electrons down the line. So this is all about an electrical charge. So if this electron leaves, the overall charge of this protein actually increases. Now remember, that's actually a loss of energy. Oxidation is lost. So when it loses energy, this higher energy over here in this protein, that's more electronegative, will bounce over there. It's, remember, positives and negatives attract, right? Opposite charge attract. So you're a good kid, right?
Yeah.
And you're probably looking for a bad boy, let's say, if I had to guess. Is that true? Is that true? Of course not. But I mean, it's an easy way to think about it.
Okay.
What else?
I'm guessing I'm just not understanding the fad. Like, where's I understood that the NAD comes from the Krebs cycle, but where does the fad comes from? It just
it does. They both come from the same place. I'll tell you what I'm going to do. I'm going to back way up, and I'm going to do all this step by step, and it'll probably clarify some points. So I'm going to walk away from the video for a moment, and then I'm going to start all the way back to the equation and work our way to this end point. And hopefully, whenever I do that, I'll do it a little slower, and it'll clarify some points. So I'm going to pause for a moment and go back to the beginning. Let's see, where are we? Okay, I'm assuming you've balanced equations in the past, correct?
yes.
All right. Well, let me see now. I need to be able to. All right. I don't want the magic pen. Hold on. Well, hold on just one sec. I need to get. I'll actually do it with these pens. You can still see it just as easy. Okay, doke. So here I've got, this is going to be photosynthesis. And what are some ways that you know that this is photosynthesis?
They're using sunlight and carbon.
Okay. Yep, you got sunlight, you got carbon dioxide as a reactant. I got water, that's a reactant. I'm producing glucose, I'm producing oxygen. So this is going to be photosynthesis for sure. Let's just balance this, and I want to stick with the theme of balance, and balance is going to help you understand where that NAD and FAD comes from. So how many carbons do I have in carbon dioxide? It's 6 molecules of carbon dioxide.
Yes, 6.
Yeah, I did have 6. Remember, assume that there's a 1 here if you don't see anything behind it. So 6 times 1 is 6. Excellent. And then how many oxygens?
12.
12, there you go. So 6 times 2 is 12. So we're done with this. We'll go over here. Hydrogen, 6 times 2 is going to be 12. Excellent. We get rid of hydrogen 6 times, assume there's a 0. That's going to add to my oxygen. That's going to be 6. So I've got 18 here. And I'm doing this with the mouse, and normally my handwriting's not that bad. But I've got 6 carbons, 12 hydrogens, and 18 oxygens. Excellent. Here, 6 times 1, 6, assuming that there is a 1 in front of this if there's no number. So 6 times 1 is 6. 1 times 12 for a hydrogen is 12. 1 times 6 is 6. So we're done with this. 6 times 2 is 12. So this is balanced. Everybody's happy here, so there's no crazy, not breaking the laws of conservation of energy or matter or mass. Everything is the same on both sides of this equation. We're okay with this. So let's go to the next. And this is going to be respiration. So I know it's going to be respiration for a different reason. So I'm going to go back here. I've got 6 molecules of carbon and 6 molecules of water. So that's a total of 12. And I've got 1 molecule of glucose, 6 molecules of oxygen, so that's 7. So 12 and 7, but the same amount of matter. This matter's more broken down on this side. So this is definitely going to be going from a state of disorganization to a state of organization. So I'm going from being disorganized to being organized. So this has got to be reduction overall, because remember, reduction is gain. You're gaining energy. You're using electrons to bond this together, this molecule of glucose. So all these hydrogens are all carrying these electrons, and that's really where the action is. So if we look at this molecule of glucose here, you can see it's gotten broken down here. So it's lost energy. I'm going from, this is 1 molecule of glucose, 6 molecules of oxygen, to 6 molecules of carbon dioxide and 6 molecules of water. So I'm going from 7 again to 12, but I'm going it backwards. So I'm going from a state of organization to a state of disorganization. So this has got to be oxidation. And we know it's oxidation because I'm using this oxygen right here to break this down. So this is where oxidation is loss. I'm losing energy overall. Their energy is getting stored right here. I may be breaking this down, but I'm taking the energy here and I'm storing it over here, but I'm breaking it down into smaller pieces. So from really 1 molecule to 36, if you look at it that way. Nevertheless, this is also going to be 6 carbons. I just want to make sure we're not lying to anybody. So this is 12, 6 oxygens here. That's 6 carbons, 12 hydrogens. 6 times 12, or 6 times 2 is 12 here, and there's 12 atoms of oxygen and 6 molecular oxygens. So that's 18, 12, and 6, and we'll do the same here. 6 times 1 is 6, 6 times 2 is 12, and stick with this theme of balance. You'll look at this, think, wow, we're doing all this. It'll make sense in a little while. So I've got 12 here, counts those out, and then 6 times 1, that is going to be 6. So I've got 18 here, so we're still balanced, and that's okay. Now you might be wondering, what about these ATP? Well, there's a reason. What about these guys? There's a reason that I'm not counting them, and whenever we get to this diagram in a moment, I'll explain why I'm not counting these. But at this point, I'm not going to be. We're recycling this, and these are coming from outside of your body, right? I get this carbon here, or this carbon-based molecule, this glucose, from eating. I'm eating this food. This is oxygen, and I'm getting that. How am I getting this oxygen again?
From the air.
Yeah, I'm breathing it from the outside, from the air. So this is coming from outside of me. And then this is from the inside of me that's going out. So like this is carbon dioxide that's inside, it's leaving my body. So this is also breathing. And this water I'm getting out by urinating. But I'm going from inside out and outside in. These are external reactants. These are reactants from the outside of the mitochondria. These are already within it, and you'll see what I mean by that in a moment. So just to remember that these equations are balanced. You can see that reactants on one side are the products of the other reactions. So here's respiration and photosynthesis. Take these stars and tell me, where should I put these stars? These stars are going to represent energy. Here? What do these represent? Yeah. What could I put those on?
I don't understand.
Well, okay, so in this case, I've got some of these molecules are going to represent energy, and I'll give you one that doesn't. So you breathe out carbon dioxide, correct?
Yes.
Okay. So your body is not like, oh my God, I'm losing something that's a high energy molecule. You just don't worry about it. You just breathe it out. It's a waste product. It's not valuable. Anything that's valuable in terms of energy, your body's going to try to conserve it at all costs. And so this carbon dioxide will definitely not go on top of this carbon dioxide because it's a waste product. So I don't really care about keeping that. What do you think your body really, and down here in respiration, well, let's start with respiration. What do you think has energy?
The ATP.
ATP for sure. We're going to use that to carry out the different bodily functions. And then what molecule are we using to get this? There's really two molecules. There's oxygen and there's, in this case, glucose, but it could be any carbon-based molecule that you eat. So which of these two molecules do you think represents energy or has calories?
Glucose.
Glucose, definitely. It's a food. It has calories. And some people last time said water, and I was like, well, water doesn't have any calories. If you get a bottle of water, look at the caloric content. There's no calories there. It's not energy rich. Well, what about up here?
The glucose and the oxygen.
Can't be the oxygen.
Oh no.
Oxygen would be a waste product too. Something else. It is not chemical energy. So this is chemical energy, right? Because we eat this energy.
Oh, sunlight?
It's the light energy. Excellent. So this is where the energy is in these reactions. So in this reaction, we're taking, energy, light energy, and we're converting it to chemical energy. And then we come down here and we break this chemical energy down. So again, this is going to be, get this here. This is going to be reduction. And then this is going to be oxidation. This is going to be a catabolic reaction. Remember, catabolic really means the same thing, or endergonic and exergonic. And this would be also an exergonic reaction or a catabolic reaction. I'm breaking. And this is going to be an anabolic reaction. A reduction and anabolic really go hand in hand. They really mean the same thing. And forgive my writing. I'm writing with a mouse. It's terrible. Looks like Mr. Leonard, doesn't it? All right, cool. Now there's two processes. Today we're not going to deal with this one. We're going to deal with this one next Wednesday. This is going to be no oxygen, and no oxygen is anaerobic. The prefix an or a means not. And you'll see this come up over and over and over. So anaerobic means not using oxygen. Aerobic is going to be O2. So this is not oxygen. We're not using oxygen for this. This is going to be aerobic. So this is going to be when we do use oxygen. And I like this diagram because it really illustrates that glycolysis is occurring here in the cytoplasm. This is where this process occurs. Glycolysis is going to be occurring in the cytoplasm. So this would be the cell membrane, not the mitochondrial membrane, but the cell membrane. You eat glucose, it diffuses through, it has to go through a protein channel because it's a pretty big molecule. Facilitated diffusion carries us inside the cell, and then you break it down into something called pyruvate. Now, if there is oxygen, this is the route you take. You go towards that mitochondria, because this mitochondria uses that oxygen, and if there's no oxygen, then you go this way. This is what will happen to you if you don't burn in a fire or something. However you die, the last metabolic reactions that occur in your cells are going to be this. And it's true for everybody. When our body when we stop breathing, our cells are still going to be living, and they're going to go, oh my God, I don't want to die. And they're going to start grabbing this pyruvate and converting it to ATP. But 2 ATP is just not enough to keep you functioning. And this is a last-ditch effort for your body to keep hanging on. Or it was also that turbo he talked about. I'm going to break this down, glycolysis down to detail first, though. So there is, and this is what we call an investment phase. So have you ever heard the expression, it takes money to make money?
Yes.
Okay, so this is what this is. This is the money investment phase. So there's used and there's formed. And this is something you actually do need to know that does come up on the IB exam. So we're going to generate 4 ATP from this process. There are 4 ATP that are going to be created here. But we're going to use 2 to do it. So I have to, in this case, think about this as money, right? So this is going to be the currency at which your body spends things. So in order to make money, I also have to spend money. So if I sell eggs, then I have to buy eggs to sell in the first place. Or if I sell eggs, I have to feed my chickens so that they generate eggs. Either way, I have to spend money to make money. So if 4 minus 2 is going to yield only 2 ATP. So you do need to know 2 things for glycolysis. You really need to know that we're going to spend 2 to have a net gain of 2. Do you know the difference between net and gross?
Yes.
Okay, so this is my gross. So this is what somebody paid me, but this is how much money I actually made because I had to spend this money here to make this money. So we net out these 2. There are some other products that are also formed here, and they're going to be important as well. We'll get to those when we get to the Krebs cycle electron transport chain. But you were asking about NAD and FAD earlier. Well, the very first NAD that we generate here in glycolysis. So we're going to generate these 4 ATP, but it's only a net gain of 2. We'll generate 2 pyruvates, and then we're also going to generate 2 NADHs. Now the next slide, when I get to the next slide, is where you're going to have to tell me where to put things. And you tell me as best you understand. And then I will, if you're wrong, I won't do it, but if you're right, I'll move it. Okay, so here we go. So I've got these molecules. Oh, I gotta get rid of my pen for a second. Okay. So I've got 3 processes. Remember, respiration happens in 3 steps. Which step is first?
Glycolysis.
Absolutely. So let's put glycolysis here. What is the substrate or the reactant for glycolysis?
Glucose.
Right. Okay. And and it's easy to remember. Remember that glucose is a sugar, and glyco means sweet, and lysis means break. So we're breaking sweets. That's literally what this means, breaking sweets. So I've got glucose being broken by the sweet break. And what does it generate? There's 3 products. There's one here, here, and here. And I'm going to give you a hint.
Okay.
These explosions represent what we generally use as the currency for our metabolic reactions, the energy that we use for most of our metabolic reactions. So what are those? Let's fill in these blanks down here.
ATP.
ATP. How many do we generate? How many do we net for glycolysis?
2.
Amen. Okay, now I've got 2 other products. What other products do we get for glycolysis?
The one to the right is pyruvate.
Yep, how do you know that's pyruvate?
Because it goes into the Krebs cycle.
You are correct, ma'am. That's right. Okay, so let's deal with this one. What's our other product?
Is it carbon dioxide?
It's not. It is not.
No, carbon dioxide. Okay.
Yeah, carbon dioxide comes from something else. Yeah.
Oh, is it the NAD?
Yeah, it's NAD, and it's 2 NADHs. So now let me ask you a question. Do you think that this is a reduced molecule or an oxidized molecule?
Reduce.
Yeah, and why do you think it's reduced?
I just guessed. I don't know.
Okay. Okay. There's a couple of ways. Overall, this process is going to be oxidation, right? So in order for me, I'm going to oxidize glucose. So if I'm oxidizing glucose, if I'm breaking down glucose, I have to be reducing something else. So pyruvate is an oxidized form of glucose. When I oxidize something, something else is being reduced. So ATP is being reduced and NADH is being reduced. Another way of knowing is that you can see that it's carrying that hydrogen. And because it's carrying that hydrogen, it's carrying what? What is hydrogen able to carry? with
Um,
it?
energy?
In the form of what? What part of the atom?
Oh, electrons.
Electrons, exactly. So it's holding these electrons. And so this is going to be reduced. Okay, so now we're cooking with Crisco. What do we have? What do we do over here? What do you think? Still a
Um,
Krebs cycle.
I guess I'll just start with the bottom. Is also
Okay, I would. I'd get rid of those. So how many ATP do you think we generate here? Do you remember?
2.
We do. There's 2. Excellent.
Um,
Okay. What else do we generate? Remember what's being used in the electron transport chain. I'm going to go ahead and drop that in there. We already know what that is.
what do we use?
I'm going to get my pen back. Yay. This is also the electron transport chain. Okay.
Okay. How many are there for the Krebs cycle?
It doesn't matter. They won't get that deep in the weeds with you. And you don't remember that he was talking about that it made acetyl-CoA? You don't need to know that either.
Oh, okay. Because
I
I
wouldn't
really
even worry about
that part.
because yeah, don't even worry about that.
Okay.
Yeah. What's your major going to be in college?
I don't really know.
You're still not sure? Okay. Well, if you're not a biology major, you probably never have to worry about that again.
I wanted it to be biology, but now I'm like, oh no.
Biology is easy. Once you get a hang of it, it's simple.
Okay.
You're plenty talented enough to do it.
Is it the fad as well?
It's FAD. So I got both. I got NAD and FAD. So we got NAD and FAD that are coming over here. Now it seems like we've got everything filled out except for one other thing. What else do we need to do?
36.
Yeah, these 32. So there's 36 overall. And that's just kind of a general number. So 2 plus 2 plus 32 is 36. That's not always the case. I mean, you don't have to run glucose through here. You can run a fatty acid through this. You can run a protein through this. You can run all sorts of biological molecules through here. It could be fructose. It could be all sorts of stuff. And each one of them is going to give you a different outcome, because each one of these has a different number of hydrogens, and it's all about the number of hydrogens. So fatty acids can generate up to 38, I mean, depending on how efficient your mitochondria are. Okay, so we still have some problems. It doesn't look like we've got some areas that we need to fill in here. So what do we do? What do I do with this other stuff? And I want you to we know that there's other things happening, but there's no places to put them. So I'll draw those on there, but I want you to tell me what do you think I should do? What should I do with these other molecules?
Carbon dioxide's released, right? From the
Yeah, and we're from the Krebs cycle. Okay, so let's go back to that. All right, so I'm going to get my pen back. So I've got carbon dioxide that's going to be released. Nice. All right. Bring my CO2 up here. What else do we need to do?
Is water also released? But
Water is also released, and from what process is water released?
Is it also glycolysis, or I don't?
It's not. That's going to be, and I'm going to walk you through it in just a second. It's going to be the electron transport chain. But where do you think I'm getting this oxygen? So I know where this hydrogen is coming from. So I have glucose, which is 6H12O6. That's carbons, those carbons and hydrogens. These hydrogens are where all the action is. They're over here, right here. This hydrogen here on NAD and FAD. That came from this glucose. I got rid of that carbon. I got rid of that through respiration here. I breathed out the carbon from this. I don't really care anything about that. But I really want those hydrogens. I pass those hydrogens here, and those get bonded to water. And then where does this oxygen here come from?
Is it just from you breathing?
Yeah, it's just from me breathing. So I'll draw an arrow here. That's my oxygen. Excellent. Now what? I've got other things going on here. Let me move this arrow. I'm going to move this arrow a little bit. Kick this out to the side. I need to do something right in here. Something's going on. There's no balance. And I want to continue with the idea of balance because you were asking me a lot about, well, what about NAD and FAD? Well, these are internal reactants. And if you go back to the title of the lesson, there's external and there's internal reactants. External reactants and products are going to be glucose. These come from the outside. My water's on the outside. My oxygen's on the outside. My glucose is on the outside. My CO2 is on the outside. But some things remain in here. So what do I do? What do I need in order to generate FADH2 and NADH? Which of these things down here do you think I need?
The NAD plus and the FAD.
That's right. So let me draw some arrows here. I told you that you needed to come to this. You're like, let me just let one kid explain this to me. And I was like, I don't think so. Not for respiration. You don't dare. Okay, so now I look pretty balanced. So I have these reduced molecules here. They're carrying energy. They're dropping off that energy here. That energy is getting stored in ATP. These are now oxidized. So they're coming back here. NAD and FAD are coming back here to pick up the energy from pyruvate to reduce themselves to be recycled again to drop off this energy. So this is just a continual cycle. Okay, so we're still not completely balanced. What else do I need to do here? So thinking about balance, what do I need to do? And make some kind of, what do they call these things? Conclusive statements, or, if then X, if X, then Y. So if I have ATP, then I need to have
ADP?
I need to have ADP, exactly. I can't get ATP unless I have ADP to generate it. So I've got that situation there. So we're going to take these,
Wait, where does the ADP come from?
It's recycled in your body as well. What's really cool about this? You're going to love this. And if this doesn't make you smile, don't be a biology major. You use as much ATP in a day as you weigh. So if I took all the ATP, that you use on a daily basis, and I just piled it up in a molecular form, I have no idea what color it would be because these things are so small, right? They're just molecules. But if I piled them up, it would be the same mass as you. That's how much of this you use in a day. But because it is a lot, but you don't have that much ATP in you. So all it is just a cycle back and forth between these. Boom, boom, boom. It gets broken and built, 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 broken and built. And that is happening trillions of times an hour in your body. It's just amazing. Because everything that you do, every thought that you ever had, every blink of an eye, it's just practically everything other than passive transport requires this. So since you have so many metabolic functions, you're constantly using it, but it's just getting recycled back and forth, back and forth. So it's pretty amazing fact. Did you smile?
Yeah.
Okay, well then there's still hope. Okay. All right. So what next?
Would there also need the ADP for the 2 ATP in the Krebs cycle as well?
Say absolutely. Yeah. There's no magic here, right? So let's get that. So I'm going to drag those.
Would the ADP be like proportionate to the amount of ATP? So like if you have 2
Absolutely.
ATP, would you, okay.
Sure. Yeah, yeah. And I'm not going to worry about proportions at this point. I'm just trying to get the idea across. Yeah, you don't need to even worry about proportions at all. The only time that you need to know anything about proportions is this last thing that I'm waiting on right over here.
Um, is it for the NADH at the top?
Yep. So we can I'm getting this NADH here. So let me get that guy. I just gave it a gender. It's a guy. Molecular people. And what else? What about this ATP? Where do you think this goes? And I'll give you a hint. Remember, we have to spend money to make money. So
would it also be for glycolysis then?
it would be. So in order to get this ATP, I have to spend some ATP. So let me, come on, get out of my way. Now, proportions, if we want to make this proportional, I can. How many ATP do we actually use to get these 2 ATP?
4.
Yeah, these are 4. So this is actually 4. So let me change this. This is actually 4 that we generate, but we spend 2. So it really equals 2. So the real reason I put 2 in here in the first place is because that's what I net. So I can put here net. This is what I net out of the process, which is what's going to be mostly important. But this is what I gross. I have seen a couple of times on different tests where they actually ask you about this on the IB exam. The thing about the IB exam versus the AP exam is they expect you to know some stuff. And this is some knowledge that they do actually expect you guys to have. Sometimes. It's not on every test, but it's there sometimes. But they always expect you to know how much ATP is generated in these different processes. Sometimes they ask you a question about the gross versus net. Sometimes.
So to clarify, we use 4 ATP, or like we generate 4 ATP in glycolysis, but then use 2 of them, so then we only get 2. Okay.
Yes, we only get net 2. Yeah, we produce 4, but we have to use 2 to gain those 4. So we net out 2.
Okay.
All right, now you can ask me anything you want. That was it.
Why? So we have 2 NADH from glycolysis, but why is there only 1 NADH from the Krebs cycle?
Oh, there's actually more. To be honest with you, I don't even remember how many. The other thing is, this is a model of what's going on. It's not really happening in a circle. This is happening just haphazardly all throughout the matrix. This is just a way for us to understand it. We know that these molecules are recycled. I really cannot remember how many NADH or FADHs that we produce through the Krebs cycle. I have long since forgotten that. It's not something that you need to remember for this test.
Oh, also in the photo earlier where you said you liked the image because it showed that glycolysis cytoplasm.
Yeah, so I should probably put that out here too. Yeah, that's important. So they test you on that. This is happening in the cytoplasm. So let me put my Mr. Leonard writing here. This is happening in the cytoplasm. The Krebs cycle, where is the Krebs cycle taking place? What part of the mitochondria is it taking place? I'll put the first letter.
The Matrix?
The Matrix. They're remaking that movie, The Matrix. Have you heard about that?
Yeah, I never watched it though. So.
Oh, you never watched the first one? It's pretty good. Came out the first year I taught.
Will anaerobic respiration then also happen in the cytoplasm?
Yes, it absolutely does. Since what's that conditional statement you were going to make?
Oh no, I was just saying since the other one happened in the mitochondria and it had the arrow, I didn't have a.
Yeah, well, and it's because oxygen's being used. I mean, oxygen,
It doesn't
it's
need
aerobic.
much.
It doesn't need oxygen. Yeah, in fact, I teach that the next class. That's going to be something that I have to teach you guys next class. And they do like to test you on that. And this is it. I got to show you this, and I got to show you this.
Okay.
And there's actually a cooler point that goes along with these that kind of gets into evolution later on.
Um, I think I got it.
Okay. How are you doing on the project?
Oh yeah, it went really good. We
I
all
did it.
turned in our individual parts because like we all each edited our own part and Uriel offered to put them all together.
Oh, good. Oh, excellent. Yeah, so which part did you have? I'm curious.
Uh, I did number 2 and the last one, the one about the declaration, I think.
Okay, cool. I'm curious to see how you took that. What was the second question? I forgot.
It was talking about why is it mutually beneficial and like what are the benefits for each
Oh,
one.
okay. Yeah, for each one of them. Yeah, absolutely. Okay, cool. Whenever I learned this, there have been certain points in my life that I freaked out and came home and really had a terrible moment. And this was one of them. I say a terrible moment. I've talked to Mr. Andrews about this, and he sees it as a good moment. And I do see it as a good moment too. But I learned about endosymbiosis and how did the mitochondria and chloroplasts get inside of another cell anyway?
Well, that doesn't give me much hope at all for my project.
Why doesn't it give you
Oh
much hope?
no, because you were just like, oh, you broke down when you got home. I was like, oh, okay.
Oh, it was a paradigm shift. It was a paradigm shift in my life. But so how did these organisms get, because they were independent, free living organisms, how did they get in our bodies in our cells, or in any cell?
The anaerobic bacteria and like endocytosis, but like
Yeah, exactly. Endocytosis. So the only way that mitochondria got into our cells in the first place was through endocytosis. So the implication is what? What's the implication?
that they used to be their own organism.
Yeah, they
Like,
were their own. Yeah, but we got that. But that's staring at you in the face. But the implication would be that I have an ancestor that was a single-celled organism.
oh, okay.
I mean, think about this for a moment. The only cells, and you may not know this, the only cells in your body that perform endocytosis to the point of eating another organism, to eating something that is literally like another cell, is going to be a phagocyte in your immune system. A phagocyte is an organism that goes through and just eats other cells. And the only cells in our body that do that are phagocytes, or immune cells. We don't descend from immune cells. Those are cells that came from another line of cells between the sperm and the egg. We grew into an organism and those cells differentiated. But for us to have mitochondria in every one of our cells, the only way that could be possible is that we have an ancestor that was a single-celled organism. That's it. There's no other explanation for it. And so when I'm sitting here and I was learning this, that isn't what I was taught, right? And I know you're Catholic, right? And what's the story that you were taught?
Oh, well, like I was taught Genesis, but like how God made us out of like clay and dirt and
Yeah. So in terms of your faith, you never consider that to be fact or dogma.
going to a Catholic school makes you look more scientifically because I was like, yeah, there's no way everyone thinks this way.
Well, as a child, baby, I did. I mean, I accepted it. I accepted it hook, line, and sinker. And so of course, I was young and had, and I had differing ideas because I had problems with dinosaurs. So it's like, well, hold on, the Earth's not that young.
Yeah.
God couldn't have created. There's a lot of things that kind of break down Genesis. But the one thing I think I held on to was that, you know, we were created in God's image, and that we sort of look like God in some kind of way. At least that was my interpretation. Maybe that image is more of a psychological image. Who knows exactly how that's taken? But I came home and to me it was questioning my faith, or questioning God and what I had been taught. And I mean, it rattled me hard. It was a huge problem for me. Endosymbiosis, I think, was probably the second hit I ever took to my faith. I had one other thing that happened that kind of rattled it, but this rattled it big time.
Oh
It
yeah, I
was,
can see how that would. For me, it was like someone said that if Genesis was true, then we would all be like related, but like incestually. Like,
well, sort of, yeah.
yeah. So then I was like, oh, okay.
Yeah, but then again, do you remember in Genesis when Cain killed Abel?
Yeah, I do remember. And I was like, how'd they find someone else then?
Well, do you remember what God said to what Cain said to God when God banished him?
Oh no, not exactly.
He said that people were going to hate him or something. So it implied that there were other people. So I think the implication, whenever I read that, I was thinking, okay, well, Adam and Eve were the first, but maybe not the last persons that God created. At least that's how I kind of rationalized that one. I
Oh
was like,
yeah,
hold
I
on a
did
second.
that part, but
I don't know. I've always been somebody that, because I'm a pretty inquisitive person, right? And I'm pretty passionate about things. And as a kid, I was thinking, oh, you know, if there's this God, then I got to know everything there is to know about him. This guy's responsible for everything. And this is pretty amazing. When I look around, I'm like, the world's pretty cool. And there's somebody that did this. Oh my God, I got to read about this some more, man. So like, I think I took it to heart, and it really meant a lot to me. And I think in still some ways it does, but you know, so whenever I was learning other things, I was like, oh, hold on a second. I don't know. This is hurting my feelings. But yeah. Don't worry about my personal emotions and opinions about endosymbiosis whenever it comes to you guys' presentations. Don't worry about that. All right, sweetheart. I've got to go to the doctor, so I'm going to let you run.
thank you.
Welcome, kiddo. Be good.